All right, DNA replication. So remember that the purpose of DNA replication is to create two identical DNA molecules from one. So we're creating an exact copy, virtually, of a DNA molecule. It's really important for cell division and cell replication that every cell must have its own copy of the DNA. So this DNA replication, this copying of the molecule, is happening all the time so that every cell in our body has a complete set of our DNA. I always talk about DNA as a zip because you have to sort of unzip the DNA molecule to access the nucleotides to do any sort of work with the DNA. So whether that be DNA replication, whether we're talking about transcription, translation, protein synthesis, in all of these instances, you have to be able to unzip the DNA molecule and access the nucleotides. So hence, I thought it would be fitting to represent our DNA with a zip. The top part of our zip here, I would like you to imagine as our sugar phosphate backbone, so the fabric part. The little zippy bits, I don't know what they're called, but the little zippy bits are basically representing our nucleotides. So they fit together really nicely, and they do so in pairs. Remember that, oopsie, can we focus? Yes, we refocus, woohoo. Remember that our bases A and T pair and our bases G and C pair together. So I haven't got the represented as 4 colors because nobody's got time for that, am I right? I'm not going to color all these in. One day, you never know. But remember that A and T pair, G and C pair. On the right-hand side of my little whiteboard, I've got the major players in DNA replication. Remember that anything that ends with an A's, or if you see this sort of word in biology that ends in A's, we're talking about an enzyme. So these are 4 enzymes that are major players in the DNA replication game. The first one there is helicase, primase, DNA polymerase, and then ligase. And we'll talk briefly about the role of each of these enzymes in this whole process. Remember that our DNA molecule exists as a double helix. So it's these 2 strands twisted together in this double helix fashion. The role of our first enzyme, helicase, is to actually unwind the DNA strand and then unzip the DNA strand. And we're going to try that. Let's see how we go. So here's our little helicase. Enzymes are really funky shapes. They're really specific. Remember that they're made up of a multitude of different protein strands, and they all bend together in a really specific, really special way. Basically, our helicase unzips our DNA molecule. These nucleotides here are now available to undergo any sort of copying. They're available to create a template for an RNA strand in protein synthesis. They're available for a multitude of things. To make a copy of our DNA, the next thing that we need to do is create a primer. You guessed it, that is the role of primase. So just like in our zips, and I have another little set of zips here, you know the little bit on the end, like the little, can I focus on that? This little bit on the end, that acts, it's like a primer for our zip. So before you can zip up your jacket, you need to connect to that little piece. It's like sets the scene almost. A primer in DNA replication is very much the same. So primase is our enzyme that creates this little RNA primer. So we might draw it like this. Oh, primase will come in and create our little primer. What then happens, DNA polymerase comes into play here. DNA polymerase, its role is to add nucleotides as we go along. A key thing with it is it only actually works in the 5' to 3' direction. That's a really big consideration for how this whole process works. So, our DNA strand here, this here is our 5' end, and this is our 3'. We know then, given that this strand is complementary, this is 3' to 5'. when we create a copy of this strand, this copy will be running from 5 to 3' in its direction. That's really important. That's perfect for us here because DNA polymerase only works in that direction. So it works from 5' to 3'. DNA polymerase can come in. It adds nucleotides as it moves along. So it literally will be going along this strand and adding nucleotides. These nucleotides, remember, will be complementary to these here. So if we had an A, we'd have a T in this strand. G, C, C, G, A, T, and so on and so forth. That's the role of DNA polymerase there. This strand is called our leading strand because it's made in one continuous fashion. DNA polymerase functions from 5 to 3'. It's our leading strand. It can just get continued, or it can just be continually made as this helicase continues to unzip the DNA molecule. It keeps functioning in that way. However, it's a bit of a different story for our second strand. This one runs 3 to 5', meaning that its complementary will run in the opposite direction. We know that DNA is anti-parallel. This one's running from 3' to 5'. That presents an issue for our DNA polymerase because it is running in the opposite direction. Ideally, it would run the same as our leading strand and just continue to replicate as that replication fork continues to open because of our helicase and create more and more and more copies of this strand. This is known as our lagging strand because it's laggy in the way that it's made. So, in the same way, our primase pops on here and creates a little primer. Just like the start of our zip, the little square bit on the end. So that's our role of our primase done. It's added a little RNA primer in. DNA polymerase then comes in and wants to add these nucleotides, but it can't function in this direction, which would be really wonderful. It only functions from 5 to 3'. So it only functions in this direction. So here's it functioning. It's adding little nucleotides in as it goes. That's that. That's okay for now when our replication fork only exists here. But remember that this process is a continuous process. It's going to keep happening. So what will happen is our helicase will actually have unzipped a little bit more. Presents an issue now for this little strand here. Our primase then needs to go back, create another little primer for our lagging strand, and then our DNA polymerase comes back and again adds those nucleotides to match up with our initial strand. It has to do so in these sort of broken fragments of nucleotides versus our leading strand, which can just continue on. Our DNA polymerase can just continue on moving down that strand really nicely in that 5' to 3' direction. These fragments that are made in our lagging strand are actually called Okazaki fragments. Oops, I think it's spelled like that. We should have checked that, but that's okay, forgive me. Okazaki fragments. What happens then, once this whole replication has happened, our DNA polymerase will come through, those RNA primers get removed, and it makes up the difference with our little nucleotides, our little DNA nucleotides. So it comes along and basically it seals up all those little gaps where those primers were. One last enzyme that functions in this process is our ligase. So it's this little guy here. He basically is like the glue at the end and comes along and tidies up any little, not so much mistakes, but basically glues everything together. So he'll run along this little strand here and make sure that all of those little nucleotides are stuck together and they've formed a proper second DNA strand, or they've really formed a proper complementary strand. So our leading side here our lagging side here, all of it gets connected, and basically, if I get my other zips, we're going to end up with 2 strands or 2 copies of the DNA molecule. So I'll just connect my little zipper in here. It will look something along these lines. All zip back up, all well and good, done and dusted. Let me just rub that out for a little second. So we end up with then 2 molecules of DNA. They're identical in their pairing, so you have still this strand being complementary to here, this strand being complementary to here, but the blue strands represent our new DNA. The white strands are the original DNA strand that we made the copy of. So the white are our conserved, because they still exist in this new DNA molecule. This whole process, or DNA replication, is known as semi-conservative because part of the original DNA strand is conserved. So it's another little important word for you. So remember that. Big thing here, DNA polymerase only functions from the 5' to 3' direction, which is perfect for our leading strand, but that means that our lagging strand, is formed in Okazaki fragments, and it's formed in different pieces. Helicase unwinds and unzips our DNA molecule. Primase adds those little primers to start the process for polymerase so that it can then add new nucleotides. DNA polymerase comes through and adds those nucleotides as it moves along from the 5' to 3' direction. And then ligase comes along at the very end and goes through it all and sort of glues it all together, zips it all up. All done, all well and good. Bada bing, bada boom.